What's up, everybody? How we doing? Today we are talking about the plasma membrane. And this should hopefully not take too long. So we're going to talk about the, kind of outside of the cell, if you remember that, if you remember what that boy is, the plasma membrane. It acts as a barrier to the cell. So things that act as a barrier don't let things in and out. So how do things get in and out of the cell? That is the question we're going to answer today, and they do that in a lot of different ways. I got a little video here at the top for us to watch. Where did that go, actually? Here it is. And then we're going to go through this. Let's just watch this video and then we'll go through that, because this is a good little video to kind of give you an overview of the plasma membrane. I really like it. It does a good job showing how fluid.
It is transparent under the light microscope and is very thin.
Resistant the plasma membrane is.
Because amphipathic molecules possess both hydrophobic and hydrophilic regions, they essentially covalently attach
so these are the polar heads of the phospholipids,
interface.
And these are the fatty acid tails.
Hydrophilic molecules always prefer to be in a more energetically favorable configuration.
Like to get away
from
water.
Solve this problem, the edges of the sheet can join with each other to form a continuous sphere. These spheres are called liposomes and can range from 25 nanometers to 1 millimeter in diameter.
Sorry, I kind of paused it there. Let me go back.
Diameter.
I meant to pause the video and I paused the video.
The entire lipid.
So this.
The entire lipid.
Dang it. In that 1 millimeter, that's the size of a frog egg, which is a single cell. And all of these, you have cells all these different sizes. So these liposomes, like I said, they're just based on the properties of lipids. Or cells size is based on the properties of lipids. Like there's nothing special about cells really, as far as the membrane goes. It's just based on the biochemistry of water interacting with the phospholipids. Millimeter big. You can see a liposome the size. You can see that with your eye. It's a millimeter. 10 of those fit in a centimeter. It's crazy.
In diameter. The entire lipid bilayer would be dragged up along with the protein. This should give you some idea of the strength of these hydrophilic interactions.
So the plasma membrane, you can really pull that thing out. Like if this was a cell, I could pull out a piece of the plasma membrane like that far, and it would just go like bloop and like snap back in there. It's really strong.
Lipid rafts therefore appear to be thicker than the rest of the plasma membrane. Having many proteins clustered together may be important for signal amplification is the presence of carbohydrate molecules on the extracellular or intraorganellar side and lipid-linked proteins that are held in place via covalent attachments. Detergent molecules insert into the lipid bilayer, and because of their cone shape discussed in a previous section, create bends in the otherwise flat plasma membrane. Also remember that the plasma membrane is fluid, so you could conceivably move the transmembrane proteins side to side within the plane of the lipid bilayer.
Pretty cool. That's important. I want you to remember that like you can really pull on these membranes, and then things are sliding around throughout it.
So you could conceive.
Pretty nifty indeed. Get that out of the way there. And so let's just go through this now. How do we get things inside and out of the cell? We went through the video. We're going to talk about glycosylated and signaling proteins, which we saw a little bit of. Then we're going to get into diffusion and talk about the different way things get through the cell. And at the end, we're going to talk about tonicity. It just has to do with salt and osmosis. So glycosylated proteins. So I've got some proteins here, just some examples. And the video briefly mentioned that. These are just proteins in the membrane. We got our phospholipid bilayer here. We've got 3 different proteins hanging out in this membrane, and they can just be on the inside of the cell, lightly associated with the membrane, or they can be transmembrane. They can go through the membrane and be kind of equal on either side. Or there's other versions too. They can be transmembrane and have bigger, more exposed areas on the outside or inside. You could have proteins like this on the outside. Just give you some different examples here. So one thing that I just want to briefly talk about quick is glycosylated proteins. Glycosylated proteins are just proteins that have sugars on them. And the video that I just showed gave an example of those as well. But these are really important. So everybody will have a kind of unique style of sugars on these proteins. And some people's are more similar to each other than others. It's kind of like a little cellular molecular fingerprint. So these would be carbohydrates on protein, which is a glycosylated protein. These are really important for your immune system being able to recognize self. So if you have, for example, a liver transplant, you need to find somebody whose glycosylation matches yours. Pretty nifty. And the other thing I wanted to talk about was just signaling proteins. So for example, sometimes things don't have to enter in the cell for changes to occur. So let's say this is a signaling protein right here. Let's say this is a signaling protein. Right there. What can happen is a molecule, a signal molecule, come in, bind to it, and then we have a signal occur. So another protein, whoops, did not want to do that. Let's say this protein here is actually usually bound to it like that. And when that signal binds, then it releases this protein to create a signal. Let's say it like clips this little part off from the membrane. So it releases a downstream signal into the cell. So nothing into the cell. So nothing has really crossed this membrane, but a signal has been transduced or crossed the membrane, even though nothing has physically crossed it. So a signaling protein, nothing physically crosses the membrane. So there's just a couple little things about proteins that I want to talk about quick. Now let's get into diffusion in the plasma membrane. And we'll go through these. So diffusion, very simply, I'm sure we understand what that is, diffusion, but let's go ahead and define that for us. So diffusion, the sciencey way to put it is something moving from high concentration to low concentration. That's the way to define it. A very simple way to think about this is, if you spray perfume, over time, it will spread out through the scent. Over time, the scent will spread throughout the room, or a candle or anything like that. So very simply, if we have a little beaker, let's say like this, and it's got some wah-wah in it, and we drop a big old hunk of, let's say, purple, crystal dye in there. Bloop, bloop. We drop this bad boy in there, like so. Just go ahead and redraw that over here. What's going to happen? This thing, over time, if we kind of zoom in on this, what's going to happen is tiny little pieces of this crystal dye are going to start to break away. And they're going to start to spread out. And I'm just going to remove those little pieces that I took. And this process will just continue on and on and on until eventually this whole thing is now going to be purple. Not in this pane, but in the next pane when I draw that. So eventually this thing is diffusing out, diffusing out. And you could run this little experiment with some food coloring in water. Pretty fun, pretty nifty. Until now, all of this purple is going to be nice and diffuse throughout. It won't be super dark. Let's see, I have a highlighter. Yeah, so now this whole thing is going to be purple. Like so. Oops. So the crystal, say like the dye spreads out over time. And this is just because of the kind of molecular movement in water, or the molecular movement in air. Spreads out over time due to the molecular movement in water or air. And so it'll just evenly spread out because it's all densely packed here. Eventually it's just going to, and then it'll be all exchanging all throughout the liquid equally. It's pretty nifty. So that's diffusion. It always goes high concentration to low concentration. High concentration to low concentration. Another term for this is it goes down its concentration gradient. It always goes down the concentration gradient. Just like if you had a lake, big lake, and it was behind a dam and you smashed the dam, the water's going to want to, it's very concentrated in the lake, it's going to want to go rush out the dam. Always goes down its concentration gradient. Okay, so that's the basics on diffusion. So now let's talk about how things get through the membrane with the process of diffusion. So we've got our cell membrane here in blue. I'm not going to draw on those lipids every time. I'm just going to draw it kind of very simply like this. So the first way things can get through, some things can slip right through the membrane. We talked about one of these, which is, for example, a steroid. So things that are hydrophobic. Things that are small and hydrophobic can slip right through. Things that are small and neutral, or small and polar. And I'll give a couple examples of these. So these are things that can slip through. So things like that are small and hydrophobic. Sorry, I spelled that wrong. Hydrophobic would be steroids. So steroids can slip right through. Bang. Things that are small and neutral, that'd be like O2, like oxygen. And small and polar, that would be something like water. So these can all get just right through the membrane. And this is why steroids make great signaling molecules, is because they can get right through the membrane. So this would be that. And this will be the same for all these. This will be outside the cell. And this will be inside the cell. All right, so this process that I just described here is just passive transport through the membrane, or passive diffusion. Passive diffusion through the membrane. Okay? Something that couldn't get through is something that's like large and charged, like a big old protein or a salt ion. These would not get through the membrane. So things like protein, big macromolecules, DNA, ions, DNA, these can't get through the membrane. Can't slip through the membrane. Too big, too charged. They don't want nothing to do with they don't want nothing at all to do with just going right through the membrane. Okay, so now we're going to be talking about through channels. So we did through the membrane. Now we're going to be talking about through channels. It's very simple. So some things we got our membrane again, and I'm going to draw a little gap. We've got some beautiful things called channels. And one of my favorites is the potassium channel, which looks kind of like a little diamond, honestly. It's actually kind of fatter at the top. Kind of drew this membrane a little wrong. It's thinner at the bottom. And in the center, it makes a nice, beautiful little channel just the size of a sodium ion. So this would go all the way around. So let's draw like a sodium ion, something like a bunch of these would be sodium ions. And they're very high in concentration outside of the cell. Those would be sodium ions. And they will just want to go right down here, down their concentration gradient to be down here. So sodium ions would love to get down here. Boop, boop, boop. I want to do those in black, actually. Come on, work with me, baby. So sodium ions diffuse down their concentration gradient through a sodium channel. And this channel is the exact appropriate size and charge to kind of attract and allow these just sodium ions to get through it. It's pretty nifty. So this here would be a sodium channel. All right, doesn't require any energy. These are both passive. So this here is passive diffusion through the membrane. This is passive diffusion through. Passive means doesn't require energy, through a protein channel. Pretty nifty. Pretty nifty indeed. All right, let's keep cruising. We've got through channels checked off, facilitated, and then active. Actually, let me pull up a little picture of these sodium channels because they're pretty cool. Sodium channel here. This is a crystal structure of a sodium channel. Two different little views here. So this would be the outside of the cell. This would be the inside. And you see this little channel here, right for these little sodium ions to slide right through. It's very beautiful. And this is important. We'll talk about why. Very important in our nervous system. Important in all of our cells, but very important in our nervous system for conducting the electrical impulse. Okay, so we've got facilitated transport. So a good example of this is glucose transport. So this doesn't require any energy. So let's do it in green. So facilitated transport, it's kind of weird. Does not require energy, but the protein undergoes a shape change. So the channel in this case, in passive diffusion, didn't change its shape, it's just a hole. It's pretty nifty. There's a lot of other sodium or other types of channels other than sodium, potassium, calcium, aquaporins for water, so on and so forth. All cool in their own regard, but facilitated transport, a little different. I'm going to draw a little membrane. I'm going to draw two little slits in this guy. An example of this is glucose uptake into our cells. Got a little fatter there, it's okay though. I could fix it in post. So with these, the basic idea is we got this. We got our protein, let's say, in blue. And let's just draw it like this. Let's draw it kind of like this, nice and simple. Easy peasy. Very easy, okay? And what happens is we're going to have a molecule, let's say, come in right here and bind. Let me draw these actually a little, something like that, and then maybe something, since this is glucose, we'll have another little thing bind. So maybe what happens, yeah, we'll stick with that. We'll just stick with that. What happens is now this protein is undergoing to go a shape change. So it'll look like this, a little something like this. And this molecule can then get through, because it's undergone that shape change, but it does not require any energy. So shape. So molecule binds and protein shape change occurs, but no energy is required. So that would be facilitated diffusion. So this is also always going to happen down, will always happen down the concentration gradient. So it would have to be, high in concentration up here and low in concentration here. Same thing, passive diffusion is also going to go down the concentration gradient. All of these are going to go down the concentration gradient. So down the concentration gradient, because it's diffusion. All of these are diffusion. Like so. Okay. An example of these is insulin uptake, or sorry, glucose uptake, which also has a co-transporting mechanism or molecule that binds called insulin. So there's actually two molecules that bind, and then we have facilitated diffusion occurring. Thought I had a picture of that to show you, but I do not. Okay, so an example of that is glucose, facilitated transport, glucose, get rid of those. All right, now we've got active transport. This one is pretty crazy. So very similar to this actually, but now we're into active transport. Active transport. which means requires energy. Requires energy. So that also means we can go in opposite. This is no longer diffusion. We can actively transport things from low to high concentration, for example. And this is extremely important inside of all of your cells, but especially your neurons. So let's just draw, I'm just going to draw this zigzag. I'm just going to copy this just to save a little time. You guys can draw this faster than I can. And what actually happens is, I'm actually going to draw it in reverse here, just to make things complicated for you. Just kidding. Just kidding. I'm not trying to make it complicated on purpose. That would be silly. Get rid of that. But I wanted to start on the one side instead of the other. Okay. Boom, boom. Fantastic. Look at that. It's like, I just did a magic trick. Okay. All right, so inside of the cell, where did I draw my sodium? I drew the sodium ions in purple. So inside of our cell, let's say we got some sodium ions. Boom, boom, boom. And we actually got a boatload out here. Boatload of sodium. And inside the cell, we've got a lot of potassium. And of course, we've got some potassiums out here. We're going to have, obviously, a couple more sodiums in here too. So we've got high sodium, and we've got high potassium. 2 S's in potassium. And this would, again, just to be clear, this is the outside of the cell. And this would be inside of this cell. All right, what I'm about to show you, 1/3 of all the food you eat goes to this. This is called the 1/3 of all the energy you make goes to this, the sodium potassium pump. So you can make a plate of food and you could just cut it into thirds, like section it into thirds, be like this is for my sodium potassium pumps and the rest is for me. That's right, you are all of the food you eat, you are feeding it, 1/3 of it, to your sodium potassium pump. All right, so what does this thing do? It keeps the inside of your cells different than the outside. It keeps the inside of your cells different than the outside. It produces an electrical charge across your cells. So sodium and potassium are both positive. But what happens, but we end up with more sodiums outside than we do potassiums inside. And so we get a positive charge outside, negative charge inside. Getting a little ahead of myself. It's okay. What happens is we're going to have 1, 2, 3 sodiums. All right. And then we're going to have ATP come in here. ATP is going to be used, broken down into something called ADP. I'll keep it all lowercase, ADP. And then we're going to have our 2 potassiums bind. So these will actually be these will be released. 1, 2, 3. These guys here will be released. So 3 sodiums get kicked out and 2 potassiums get brought in. And I actually need to reset this guy like so. So that we have a third little version of it. So then it gets reset. And these would be then released. All right, so let's walk through that again. I'll verbally explain that with some arrows too. So these guys, 1, 2, these guys are all coming in. Let me get rid of the arrow except for the last one. So these guys are all coming in here. This looks like trash, Tori. All right, so all three of those are going in there. Looks like garbage. It's okay. And then they are all being released. Boop, out here. Let's say. While these guys, whoop, are both coming in there. That's the better way to do it. Let me fix this. It looks like garbage. It's bothering me. Oh, that's now we're talking. Yep. But it looks like an Atari symbol. Okay. Those get released. These guys come in, and then we've got our two sodiums, or sorry, our two potassiums coming off. Boom, boom, right? So these guys came off from right there. Don't want that. Want it thin. Don't want the arrow. But, okay, those guys come off. So we get 3 sodiums get kicked out, 2 potassiums get kicked in. So the sodium-potassium pump keeps our cells different from the outside, inside different from the outside. So 3 sodium get pumped out. Yeah. And 2 potassium get pumped in. 1/3 of all your energy is going to do that. All your cells do it, but it's very, very important in your neurons. And let's see. And that requires ATP, requires energy. I got a little slide here to show you. Because we got a little Drake here doing his little thing. And this is the mnemonic that I used to remember this. I literally use this. I didn't create this GIF, but I created this mnemonic. Drake is a bouncer. A mnemonic is something to remember. Drake is a bouncer at a nightclub during ladies' night. Okay? What happens on ladies' night? You got too many dudes in there, you got to kick them out. You got to kick out 3 dudes for every 2 ladies that you bring in. Nah, go outside the cell. Nah. Oh, 2 ladies, you want to come in? Okay, come on in. That's just the code. That's just the way of the bouncer Drake on ladies' night. And this is how it works. So we got sodium, potassium. Right there, we got boom, boom, boom. Sodium's hanging out. Potassium's hanging out. Well, this is how the process works. So 3 sodiums out, 2 potassiums in. Don't forget it. Maybe on the test. Okay, we did that. Active transport, sodium, potassium pump. 1/3 of all your energy goes to that thing. Pretty crazy. Okay, other couple little things I want to talk about real quick is endocytosis and exocytosis. I'm just going to draw one membrane to do this quick. Endocytosis and exocytosis. This is a way to like move big things in and out of the cell. So if you remember, we actually talked about this already. We actually 100% already talked about exocytosis. So this is the idea of fusing a vesicle to the membrane, and then you could dump the contents out. Endocytosis is the exact opposite. So we'll draw those out. So endocytosis and exocytosis. Draw these here. So this is again the outside. This is the inside. And let's say we got some, we got some like little red goodies out here. Like so. Like that. Well, let's say this cell wants some of these red goodies. What it can do is it can start to pinch off a little bit like that. And then over time, what'll happen is it'll pinch it off even more, like so. And then eventually, it'll just be fully pinched off. And this would, of course, be full of our little red goodies in here that it swallowed up. And now it's got its little red goodies in it. And it can take these and fuse them with a lysosome. If you remember what that is, go back here. So our lysosome is the stomach of the cell. It's got an acidic pH to digest stuff. So this is a way that cell can bring in a vesicle and shove it in that stomach to digest it. Pretty nifty indeed. All right, back there. Okay, so this would be endocytosis. Endocytosis. And then we got exocytosis, which is the exact opposite of this. So if we just keep kind of drawing this out, and let's say we've got a little vesicle, and this one's got purple gunk in it. We want to get rid of it. Well, this thing's just chilling. This thing right here just chilling and get closer. Boom. Gets closer. Which actually I just like these things. They're called snares. I talked about them already. They're like little molecular threads, and they actually connect to the membrane of the vesicle and the cellular membrane. They kind of intertwine. And then they twist, like twist ties, and they cause a fusion to occur between the vesicle membrane and the plasma membrane. I already talked about this. Looks something like that, and then it dumps out the contents, and then eventually you just have a normal looking plasma membrane with the contents outside the cell. Pretty cool. So this process would be called exocytosis. This way, really big stuff can get in and out of the cell. This would be exocytosis. So just to be clear, these are like little transitions. Go from that to that to that. This is endocytosis, and then this to this to this, and this is, actually I'll kind of do it like this. This is exocytosis. Boom. Great. All right, last but not least, tonicity. What does this mean? What is tonicity? What is this you speak of? What is tonicity? Tonicity has to do with a tonic. Anytime you hear that word, let's talk about it, huh? Tonicity. Let's talk about it, huh? Tonicity? This just means salt. Just to give it a salt. So this is salt. This be how salty is your water. Okay, so we have a couple different versions of this. We can have right in the middle, we got isotonic. Iso means the same. Isotonic, it's the same salty as your cells. The same saltiness as your cells. That's what isotonic means. We can have something that's really amped up. When something's really amped up, that is hypertonic. This means more salty than your cells. And last but not least, we got hypo. That's low. Hypotonic. What does that mean? Less salty than your cells. Okay, so now we're talking about the solution. We're not talking about the problem, we're talking about the solution. You can talk about the tonicity of your cells too, and it would just be kind of the opposite, but I don't want to get too confusing here. Let's just stick with this. So solution. The solution. Solution is hypotonic, isotonic, or hypertonic, depending on how salty it is. So let's say, I'm trying to remember what the actual tonicity is of our blood. I think it's 0.9. Doesn't matter. We'll figure that out in lab. I can't remember off the top of my head what it is, but let's say, we'll just do it like this. All right, so we got our little beaker. We got our little beaker. And we got our last but not least, our little beaker. We got our water. Okay, so this is going to have a low, this is going to be low salt concentration. Low salt. Actually, yeah, I'm just going to draw in the salt actually, like this. 1, 2, 3, 4. Very low amount of salt. Isotonic, we're going to draw it with 8. 1, 2, 3, 4, 5, 6, 7, 8. 8 salts. And hypertonic, we're going to go crazy with 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. So a lot of salt in there. Okay, these little things are salt. All right, now let's draw in our cells. We're just going to draw in normal looking cells to start. Don't actually draw these. Not yet, but I just want to kind of explain what's going to happen over time. So we got a cell there. We got a cell there. And we got a cell there. I'm going to draw them in, but I'm going to change them. We drop those in. What's going to happen? All right, well, let's draw in our salt here. So our isotonic, it's going to have 1, 2, 3, 4, 5, 6, 7, 8 salts inside the cell. 1, 2, 3, 4, 5, 6, 7, 8. 8 salts. Okay, this one's going to have 8 salts. 1, 2, 3, 4, 5, 6, 7, 8. And this one is going to have 1, 2, 3, 4, 5. Or sorry, this one's only going to have 8 too. 1, 2, 3, 4, 5, 6, 7, 8. All right. Right. Cool. Okay. Well, this hypotonic solution, water always follows salt. I'm just going to write that down. Very key. Water always follows salt. Water always follows salt. And we'll actually start, we'll start here. So here we have 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2. We've got the same amount of salt inside and outside of the cell. It's at the same concentration, even though this seems a little higher concentration. Let's just say it's the same. So what this means is that water is going to evenly enter and exit the cell. So we're going to have the same amount of water entering and exiting. Let me draw this a little more clear. So it's the same amount. Same amount of water. We'll just call that H2O, like that. H2O, H2O. So it's going to be the same because there's the same amount of salt on either side. Whereas this, we've got a lot of salt in here compared to out here. So we're going to have way more water trying to get into the cell. Right? And only a little bit trying to get out. So H2O, I'm not going to draw all of these on. H2O. Way more trying to get in than trying to get out. And what this is going to cause the cell to do is, I'm going to delete this now. The cell will swell. And if it's really going to swell, it'll actually burst. So the cell is going to swell way up. Get way bigger, and if it's in very, very low salt water, it will actually break open. So the cell will swell and potentially burst because it's going to fill with water, and it can only hold so much water. It's going to fill with water. Okay? So then what do you think happens in a hypertonic solution? There's way more salt out here, so we got water is going to end up leaving the cell because it wants to follow salt. Boom, 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 boom. Water would much rather hang out there than go into the cell. So what's going to happen here is the cell is actually going to shrivel up. It's going to get smaller. So here the cell will, it's called crenation, will actually start to shrink up, and shrivel up like so. I got to keep these little salts in. Cell will shrink. Cell will shrink. It's called shrink and shrivel because the water is leaving it. It's getting less fluid. It's less full. Crenation. Crenation. Pretty nifty stuff. Okay. So this process, we're going to explore this in lab actually, a couple of different ways. This process is called osmosis. What is osmosis? It's a very specific thing, and I'm going to put this in red because it's really important. Osmosis. What does that mean? Osmosis. Osmosis. What was diffusing across the membrane here? It was water. Osmosis is the diffusion of water across a membrane that only lets some stuff cross it. Another way to phrase that is across a semi-permeable membrane. Another way to just phrase this is another phrasing, more sciencey, is the diffusion of water across a semi-permeable membrane. Frickin' wonderful. There's often a misguided kind of joke saying that if I just sleep with my head on my biology textbook, I'm hoping to learn through osmosis. You can't do that because osmosis is the diffusion of water across a semi-permeable membrane, not knowledge from a book to a skull. So it's actually pretty cool. Like, let's say we take a little beaker, and that's what's happening there is water can get through here, but salt can't. Salt can't just diffuse across the membrane unless there's a channel. Because, why? If we go here, it's ion. Salt's an ion. It can't slip through the membrane. The membrane's semi-permeable, and ions can't get through. Both salts are ions. So we would need a sodium or potassium channel in order for the water to slip through. Okay. So the diffusion of water across semi-permeable membrane, and where was I going with this? Yeah, this is pretty cool actually. So if we take our little beaker, we got a little little glass tube in there that is something like this. Let's say maybe it's got like a little, like a little bulb up here, or actually. Little bulb would be down here, let's say. Doesn't actually really need a bulb, but it's cool for effect. And then we got our liquid. We got our liquid water here. And let's say this is pure water. Pure water. Pure water. And this is like there's a little tiny membrane here that we've added across the bottom of this. Let me draw that a little fatter. This would be a semi-permeable membrane that's permeable to water. It's permeable to water, not salt, let's say. And then we got a little bit of liquid in here. Actually, probably fine that way. We got some liquid in here, and it is going to, I'm going to draw in yellow because it's really, really, really high in salt. This thing's full of liquid, and it's really, 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 really high in salt. Let's say it's like, I don't know, super salty. Let's say it's like 30% salt solution. What's going to happen is that over time, basically the liquid volume in there is going to raise way up. I'm just going to recopy this. Just to save a little time here for myself. What's going to happen is this fluid in here is going to raise way up because the water is going to rush inside. So we're going to have water rushing inside. And so we can actually have water defy gravity. It's pretty nifty. So water will defy gravity because osmosis is such a strong force. Such a strong attraction. Because the water wants to dilute the salt and make the two sides equal. It's basically just a specialized form of diffusion. Pretty nifty. We're gonna play with that in a couple of different ways. We're gonna do it with little bags, as well as look at cells and a lot of cool stuff. Very exciting. All right, I think that's all I wanted to cover today. Osmosis, very important. It's very different than just diffusion. Osmosis has to do with the movement of water. Diffusion is the movement of anything. All right, we did it, and I even put in some extra stuff. All right, I'll see you guys in the next one. The next one's also pretty quick. And then that's it for the lectures this week. Yay. Good luck on your exam.